DNA replication in prokaryotes. Separation of two complementary DNA strands. Replication begins at a single, unique nucleotide sequence known as the origin of replication. A replication fork forms and marks a region of active synthesis. Replication is bidirectional with a leading and discontinuous lagging strand. Leading strand is replicated in the direction in which the replication fork is moving. Synthesized continuously. Lagging strand is copied in the opposite direction of the moving replication fork. Synthesized discontinuously. The short, discontinuous fragments are known as Okazaki fragments. RNA primer. The RNA primer is made by pyramase, necessary for DNA polymerase III to initiate replication. Chain elongation. Chain elongation is catalyzed by DNA polymerase III, which has both polymerase and proofreading functions. Elongates the DNA chain by adding deoxynucleotides to the 3-hydroxyl end of the RNA primer. Continues to add deoxynucleotides from 5 to 3 direction until it reaches the primer of the preceding fragment. Proofreads each newly added nucleotide has 3 to 5 exonuclease activity. DNA polymerase I degrades RNA primer, has 5 to 3 exonuclease activity. DNA ligase. DNA ligase seals the remaining nick by creating a phosphodiester linkage. Overall, eukaryotic DNA replication is similar to that of prokaryotic DNA synthesis, with several notable exceptions. Namely, begins at consensus sequences that are rich in AT base pairs. A karyotic genome has multiple origins of replications. Eukaryotes have separate polymerases for synthesizing RNA primers, leading strand DNA, lagging strand DNA, mitochondrial DNA, and DNA repair.